Welcome everybody. Good to have you on board today. West Lab really enjoys these lunchtime discussions with you about topics of mutual interest. Now today we have a topic which connects with our innovation pillar. Australia is a country that stands in the global forum alongside America and the UK, and very, very innovative on the cutting edge of science. And so any Australian distributor needs to be nimble and needs to be ready to move on that cutting edge. And one of our aims as a company is to move on that cutting edge. So innovation is one of our core pillars. In the education space, we have taken note of the fact that there's a lot of government investment in STEM spending to encourage students to explore science and to enthuse them to get involved in scientific research. So we're looking around for products which we can introduce to the education space, which will stimulate that investigative edge of STEM programs. We had one innovation last year which we added to the range, and we're looking for more innovations, so please be free in the chat to suggest products and areas of focus for the future development of your projects. Last year we introduced the Bento lab, which is a portable DNA laboratory. It's the size of a laptop, and it's got everything that you need to get a DNA footprint of whatever you're analyzing. So the message we want to get through is that we're interested in enriching your STEM programs, and not only at STEM schools, but across the board in secondary education. I might also mention that we are involved in health, analytics, food and beverages, and also pathology, so that a lot of the products that we source and chemicals, for example, we can give you the benefit of the bulk buy in the education sector. Now today we have invited Erica Trickett from Monash School of Science, who is the laboratory technician there. She's very, very motivated and passionate about science, and they have some very, very good students coming through that school. Erica has been using the Bento lab for some years, and so we've asked her to come on today to demonstrate how she uses that to supplement the skill curriculum at John Monash. She's the laboratory technician there. She's been working at the school for more than 6 years. She has a diploma and associate degree from Box Hill in biotechnology, and she's currently studying for her Bachelor of Science at Monash. So we'd like to welcome you, Erica. Welcome to the webinar.
Thanks, Niall. So what I'm here to talk to you about today is the biotech applications of the Bento lab. Some of the major applications that we use it for here at John Monash Science School is in year 10 biotechnology units, and it can also be used for forensic units. But another major part is the VCE Unit 3 biology curriculum, where there's been a major shift in biotechnology of all facets into that curriculum. So we use it in the year 10 unit quite a lot with the emerging field of in biotechnology. So the students, they learn about all sorts of different processes within that can be done on the Bento lab. But just for your background, some of the interesting things that can be done with a piece of equipment like that is, or some things that have been done in the past with this sort of equipment include COVID diagnosis testing. Now that was done using PCR, and another massive one is barcoding of plants, which is done using PCR, electrophoresis and sequencing techniques. And then the most recent one is the acquitting of Kathleen Folbigs yesterday. Now that was done using genetic testing of her children, which is also PCR and gel electrophoresis. So it's all relevant to current stories and current research that's going on in university. Yep. So when we used the Bento lab at John Monash Science School, we originally purchased it in 2021. So during the COVID years, but that doesn't mean we didn't use it. So we use it quite a lot for student projects, specifically in the school. It's easier just to have the one unit to give to a student rather than giving them all the bulk of equipment that I have upstairs from other companies and things like that. We use it in conjunction with the Bento reagents and a new barcoding kit that is due to come out very, very soon. Some of the beautiful features include the gel electrophoresis tray, which is the orange lidded section there. We've got the centrifuge in the middle, and we've got the heating mantle, the heating block, the thermocycler on the far right. So yeah, it does. It's got the centrifuge. It's got the PCR and then the gel imager as well.
Might just step in here and say Bento have got a beautiful website, bento.bio, which has got a lot of resources, so that if you are using the Bento, there is GIF images on there showing how to pipette properly. There are experiments that you can formulate, and it's got all the experiment sheets. We will also be providing you with an experiment sheet for today's experiment. And as Erica mentioned, we will be putting together a kit for the Australian curriculum. We'll advertise that in some of our future promotions. But this is a site worth looking at, and she's done a lot of work, and so it's available as a teacher's resource.
So now I'm going to show you how we actually use the Bento lab. Now, I did these demonstrations a few weeks ago at the West Lab Experience Centre here in Melbourne CBD. And you'll see parts of the Experience Centre in the videos while they're running. But the first video is using a really simple extraction, dipstick extraction kit. And now it can be used for fruit, plants, all sorts of other weird and wonderful samples. Being in Victoria, though, we're only allowed to do extractions, DNA extractions of plant materials for further research purposes. So in this experiment, you'll see me chopping up an apple. I've successfully done it before, though, with a spring onion, with mushrooms, with all sorts of berry fruits, so bananas, blueberries, kiwis, and the PCR, like it goes straight from the sample into the PCR. So it's so simple. We can move on to the video.
We're here today to talk about the Bento lab, which is a all-in-one thermocycler, gel electrophoresis and centrifuge unit. So here I'm going to extract some DNA from an apple, and I'm going to use the very simple DNA extraction method using a dipstick. Just chopping up apples. So we chop the apple into very fine pieces. And I'm going to pop a small sample into the extraction. and then we place a small amount of the fruit into our extraction buffer. Next we take a micro pestle and we crush the apple into 500 microlitres of extraction buffer. We have to crush for approximately 2 minutes. Okay, I'm going to do the dipstick. Next we take our dipstick, which has filter paper on the very end of it, and that's what absorbs the DNA. So we dip our dipstick into our extraction buffer, ensuring we don't get any excess cells. Then we dip it 3 times into wash buffer. Next we place it into our PCR reaction mix, which includes master mix, primers, water and our sample DNA. I like to cut off the little end of filter paper just to ensure that there is enough DNA in the reaction. It just guarantees that there's DNA in my sample. We place our tubes into the thermocycler and press the lid down and turn it to thermocycler setting on the Bento lab. And we press the PCR cycle, which gives us 3 cycles of temperature, including 95 degrees denaturing, 56 degrees annealing, and 72 degrees extension. This procedure goes around 30 times and takes approximately 2 hours to complete in the classroom. Next, the centrifuge, which we can have up to 6 samples in our little centrifuge, and it has a little interlock. So while it's running, you cannot lift the lid. It runs at approximately variable speed of up to 8,000 Gs. And you can also get little adapters to fit smaller tubes. This centrifuge is set for 2 milliliter tubes to 1.5 milliliter tubes.
So let's move on to making our gel. Now, what we've got here with the arrow is the electrode. So we'll see in a minute that I have to put certain things into this gel tray to make it to protect the electrodes, and this just means that it's an all-in-one inclusive unit. So there's no mess. It uses low volumes, and we just have to remember to add all of our buffers to the gel before we pour it in. We can go to the video.
So now I'm going to make a gel electrophoresis gel in the Bento lab. This is done by adding the dams to the gel tray. Now there are 2 dams, one on each end of the electrodes, and these are used to help protect the electrodes from the agarose gel, because we need a buffer bridge between the electrodes and the gel. Next, as you can see, I've added the comb. Now there are 2 combs that come with the Bento lab. There's a 10 well comb and there's a 12 well comb. Next we're going to make the gel, which consists of 50 mils of 0.5 times TBE buffer and 0.5 grams of agarose powder. We need to mix them together and then we microwave them to dissolve the agarose. When we're microwaving, we're effectively trying not to boil but just dissolve the agarose gel or the agarose powder. So in doing that, we need to get it to almost boiling temperature. And this gets quite hot and the glass bottle does let steam out. So we have to be really careful when we're handling hot agarose gel. You can see here that the gel is now nice and clear. There's no crystals. You can see it's got like a thicker consistency than water. Next we're going to add a stain to ensure we can see our DNA samples. Now this is called CyberSafe. CyberSafe allows the DNA to fluoresce under UV light. And we're going to add 5 microliters of CyberSafe to our agarose gel. To mix, we need to swirl the CyberSafe and the agarose together. Now you can put your gel aside and allow it to cool. Done. Once the gel is at 50 degrees Celsius, we can pour it. It's still liquid at that temperature, but you can handle the bottle. So we pour from the bottom and allow the gel to move through the tank. And we pour only to approximately the 10 milliliter line.
Pipetting. Now, when you're working with gel electrophoresis, pipetting skills are of the utmost importance because we need to make sure we don't break open the gel, especially when we're putting our samples in. We have to make sure we're getting an accurate sample size into the well. So it's quite technical for students, especially, to make gels. So I often recommend that teachers run a free class where the students learn how to pipette into a gel. Contrasting colours also helps make putting samples into your gel a lot easier. So if you've got a white bench, you can see where the wells are, or if you've got a black surface, that helps as well. Yeah, so let's move on to the last video where we show how the gel electrophoresis, how I load and how it runs.
Okay, so now we're going to set up the gel in the gel electrophoresis and get it ready to run. So first off, we have to take out the combs and the dams. So to do that, we need to lift out our dams and our comb. I lift them out straight up to ensure that I don't break the gel inside the tray. They can sometimes be a little bit tricky, but that's okay. Next, I'm going to pour some TBE buffer into the gel tank. Now this is full of free electrons that assists in moving the current from the black end to the red end, which is the negative to the positive. We need to make sure that the entire gel is covered in buffer, but we can't overfill the tank, so we only fill to just below the max line. And just remove any air bubbles that we see. So this is me unloading the gel now.
Yeah.
Right. Next, we need to add our samples to the wells. And to do this, I take a 2 to 20 microliter pipette and I take 10 microliters of my PCR sample and very carefully pipette it into one of the wells in the gel. As you can see there, the green PCR buffer. And I like to anchor my arm when I pipette into a gel by holding the elbow of my right arm to steady myself when I'm pipetting. You don't want to go too deep into the well either, because you may break the base of the gel and then your sample will not go through the gel as it should. Now I also like to add a DNA ladder which is it's a ladder that has known lengths of DNA and is used to compare your PCR samples with known base pair lengths. We slide the lid on and ensure that the black and the red electrodes are connected nice and tightly, and then we place the red and the black electrode cords into the Bento lab. Next we go into the gel electrophoresis setting. We can change the voltage and the time that we'd like to run our gel for. As you can see, I've set this one at 60 volts to run for 30 minutes. And once we press start, all we're looking for is bubbles in the top to ensure that the gel is now running.
And so what's actually happening there with the bubbling?
So what it's doing is it's running a current through the gel, and the gel is like a matrix, so it separates out the lengths of DNA, the different lengths of DNA that's in the gel. So you've got move faster through the matrix at the bottom, and you use a ladder, which is the purple thing that I just added, to estimate the length of your strand of DNA.
Okay.
Yeah.
Yes.
So it's a very good system. And it's good that you can get it in things like this, because this is more affordable than what we have at school at John Monash.
What do you use?
We use enriching e-gels, which are like a cassette that you just slide in and load. They're really handy and really quick, but they're about $15 a gel, whereas this gel costs like not a lot, like $1.50. So yeah, it's just cost comparison.
Yeah.
Use the UV light to see if
Just turn the light
the
on.
sample is moving from about, you can see some 20 minutes in.
Fluorescence in that last well
This
there
just
at the moment.
assists with ensuring that there is separation occurring with your DNA ladder, as well as you can see your samples moving, your PCR samples moving down. So the sample that I just put in will start to move down.
Okay. Yeah.
I can see some of the samples starting to move down now. Just below that well there's a little green line. Time. And you can see results, like half an hour of run time, and you know, you could be doing theory work in that period. So once the gel has completed running, we can see that there is movement of the dyes in the PCR samples. But then to see the DNA, we need to turn the UV light on. We need to protect our eyes with a viewing box or the orange cover. And we can see that the DNA has separated very nicely.
Okay, so I'll just explain the results that were just shown in the last video. Basically, sample 1 was a banana sample, and sample 2 is the same, and sample 3 was actually spring onion. Sample 4 is the DNA ladder I just spoke about. Now, the way that you count the ladder to determine your sample size is you start at the bottom, and usually the lowest strand is between 50 to 100 base pairs, depending on which ladder you've got, and then you count upwards. So I think this one on the sample 4 is a 100 base pair ladder, so 100, 200, 300, 400, 500, 600. This sample here is about 700 base pairs, and the banana is about 600 base pairs. So yeah, the Bento lab is just a really easy, simple way to get everything, all your PCR work done, all your biotech work done in one unit. Yeah. So I think we'll hand back over to Niall.
Yes, fantastic. Thanks. That was very instructive for many of our audience. It just simply shows the probably a technique that you know very well, but just how it's done on the Bento lab. And there are some pluses along the way. There's maybe a couple of ways in which you might want to implement this. You might want to have one student using the unit at a time, or we have also included in the range extra gel boxes, which you'll see in a minute, so that if you wanted to have multiple students making up their gel tanks and then just run them through the machine to actually get a result, it is, as we heard on one of those videos, actually a really cost-effective way of having this hands-on experience and doing all these processes and getting an appreciation of the skills involved. So what I'd like to do now is just advance this slide. I'd like to introduce you to a team that you probably know fairly well. And while Will is just chatting to us about a few innovations in the education team, Will Crick is our secondary education specialist here at WesLab and works alongside of Mark Burke and then the customer service team, Rochelle Crick and Jessica Thomas. Any questions that you put in the chat, we'll just process those now between Erica and myself. And in the Q&A at the back end, Erica will give you some live immediate answers to some of those questions. If there are any questions you haven't yet asked, please be free to put them in the chat now. So I'll hand over to you, Will.
Thanks very much, Noel, and thanks very much, Erica. Really appreciate it. Very good insights. Sure everyone's really benefiting from it. Yeah, thought that we just as we give Noel and Erica a little second to run through some of those questions. Also, as just a second what Noel said, it's a great time now just to put any of those questions in the Q&A area. Thought we'd just run through a couple of new features that we've added to the website and that will be coming out over the next few months. So to start with, first one is the new WesLab reward points, as you can see on the screen here. It's probably the latest addition to the website in terms of ordering. So the beauty of this one here is anyone can take advantage of it. There's no anyone that has a website login to the WesLab to their WesLab account can earn points. It just happens automatically. With every $1 spent on the website, you earn 1 point. And quite simply, those points can be used down the track to get a discount initially on the product. So they can be used at any time. There's no minimum number of points that you need to be able to use as a discount. Just any order you put in there, if you feel like using them, you just select to use those points, and it will just take that amount off the order. I would just say as well, if you don't have a website login at the moment, please feel free to jump onto the WesLab website and inquire about one, or email sales@weslab.com.au, and our customer service team will be more than happy to help out. Moving on really quickly, I thought I'd also mention really quickly the WesLab 30 Days of Discovery that we're running at the moment. A lot of you would have seen this here coming out on via email. You would have seen it on the website. So with this one here, it's sort of in conjunction with WesLab's 30th year anniversary. So yes, that's right. WesLab is 30 years old this year. Been servicing these schools for a good majority of those 30 years. So been in this industry for a while, and to be honest, this is actually our biggest deal to date. So it includes a lot of the most popular deals that we have ran throughout the past few years, those being the buy two, get one free trolley deal. There's balance bundles, there's microscope bundles, there's safety cabinets that are discounted. So definitely jump on there and have a look. We are releasing a new one every day. So yeah, keep your eye out for that. As you can see here, following this webinar, you will receive an email. Now, the email will have a couple of things on it. One being a gift card. Now, it's a voucher code to be used online. And that there is simply for anyone that's actually jumped on the webinar today. So that there, keep your eye on the inbox for that there. Maybe if you can just go to the next slide, Noel. Quickly. The email will also include the full Bento range and electrophoresis range. So that this will come out in an email, and there will be a slight deal on those ranges. So just following on from today's webinar, you will be able to get a deal on either the electrophoresis range or the full Bento range. Now, the Bento range will obviously include the Bento Lab Pro, which is the mobile genomics kit, as we've seen. It also includes the Bento Biotech kit, which includes everything you need to run the experiments, as Eric has just shown us. And also the additional gel boxes that Niall had mentioned as well. So if you're running multiple at the same time, you can get all that there, and that will be shown on that email. So keep your eye out for that. And as I said, as we mentioned earlier, the electrophoresis lab station will be on there as well. So that'll be a really good deal to keep your eyes out for. In saying that, there, I will hand back over to Niall and Erica on just to obviously have a look at and answer some of these questions that have come through to the Q&A. Thanks very much.
Thank you very much, Will. So just before we open up the Q&A, just to mention that the website orders that are placed on the website do come freight free, which is the reward for placing orders on the web. We'd really like your input as to innovative ways to use the Bento, and please feel free to email us or even put that in the chat, ways in which you develop your program to use the Bento in schools. And the other thing that we're also really interested in is you sending us in suggestions for innovative ideas that you would like to develop in your STEM or science courses, which are enrichment items that you'd like us to look for equipment to support you in your endeavour to encourage students to get enthused about science. So, Erica, we'll hand over to you with the Q&A. I trust you've got all those questions there.
Yep, there's a few questions here. So the shelf life of the chemicals is usually about 12 months in the freezer, but I tend to use mine for 2 to 3 years. It's just they don't tend to break down as quickly as they should. I think the second question is already been answered. So the cost of the unit. So I think that's fine. Can you remelt the gel? So the third question there. So what I tend to do is I make just enough gel to pour into the gel tray. So it's between 30 and 50 mils per gel, depending on how good your measuring cylinders are. So I always do tend to make them up fresh, just because it's a bit nicer, and then you're absolutely 100% sure that the cybersafe hasn't degraded in the fridge overnight or over a week. The fluorescence system in the Bento lab is amazing. The little gel imager that I put over the top of the gel is really nifty, and with the UV light actually in the Bento lab, you just have to put that little box over it and everybody can peer through and see the fluorescence. You don't need a dark space. You don't need any other equipment. It's so nifty all in one. And yes, the last question, you can absolutely do it in multiple days. So the way we do it here is I tend to do the DNA extraction in one day, and then into the PCR. We run the PCR outside of class, and then the next day we go through and we do the gel. We run the gel electrophoresis. So it makes, or in a few days' time, depending on when your classes are. So you can put your PCR samples in the freezer and store them for up to 6 months. So there's no need to worry about any of that. So it works brilliantly in that sort of respect. So any more questions? Yes. If a school wants to set up a lab for DNA analysis focus and previously does not have any equipment, how much would a setup cost? I think that's so you'd only need one Bento lab, maybe 2 gel trays, and then you'd be set up for a whole class of 20 students, 26 students. You wouldn't need to worry about buying multiple Bento labs because it's all in one thing. It's really quite a brilliant setup. You can have multiple samples running all at once. So yeah. And then, yeah, my TBE also solidifies. I tend to heat it a little bit to bring it back to a liquid, but don't boil it, or add a little bit of sodium hydroxide to it to get it back into solution. Are there kits for forensics? We're working on kits. Was there any other questions, anybody? Or are we still madly typing? They're still coming through, so I'm not sure.
That's good. We've got a bit of extra time despite our slightly stuttery start. We seem to have got extra time at the end of this, so please keep the questions.
Yeah, keep the questions coming.
We'll even close early.
Yep. Cool. Well, I think any sales questions should go to Niall and William because I'm just tech. I don't know anything about sales. So if you guys could happily answer those. I think you've answered most of the questions anyway.
Yep. Will, is there a question there that?
Yeah, there's 2, I think.
Do you want to come in and answer them while everyone's on webinar?
Yeah, absolutely. In terms of cost of the units, they were shown on the earlier slides. So it's about just under 5,000, so 4,900. List price, as I mentioned, there will be an email going out following this meeting, this webinar. So you will see us. There will be a special offer then. Probably a great time to take advantage of it. So that price will be sent through early, shortly. Sorry. But yeah, as the list price, it's around about $5,000 for one of the Bento. So the actual genomics kit itself is about $4,900. I think there's a couple more units, a couple more questions that just popped through.
Yeah. So the setup for 20 students, you would just need 20 of the little mini PCR 200 microliter PCR tubes. And you just need 2 gel trays to run the 2 gels with the 12 well cone, and then you'd be well set up to do 20 samples in no time at all. Is the unit robust? Yeah, it's relatively robust. I wouldn't be taking it out into the environment. I would take it into another lab or into like a well covered space to do work in, but I wouldn't take it outside and do work like on a beach or anything like that. I don't think it would survive the beach. And there's another question, can you show the slides with the kits and prices? I think you better wait for that email to come out maybe. Oh, here we go, we're going back. There we go. We're still working on the kits as well. That'll be coming out very, very soon.
Yep, we are developing a kit, as we said, for the Australian curriculum based experiment we saw today, which I think one of the things you said to me, Erica, was pretty much 90% of the time, maybe even higher, you get a good result. So it's disappointing if you do all that work and you don't get a clear result. So
yes, you can use kits from other suppliers. It depends what you've got in your freezer, I suppose. But like if you are starting from scratch, the Bento Lab kits are really well, really good, and they work really well. I've been using them here for a little while. So, yeah.
All right. Well, it looks like we've got time together. Thank you very much for sacrificing your lunchtime. Trust you found it worthwhile and interesting. Please let us know anything we can do to stimulate your science program and source new equipment that's coming onto the market. We like being on the cutting innovation edge, and just in health, recently we brought in vape detectors for school ablutions, which is a cutting edge program of the New South Wales government, and probably going to extend to deal with the vape pandemic that is affecting young people. So we like being part of the, bringing in new equipment to keep us moving forward. So look forward to supporting you in your endeavours too, and inspiring students in science. Thank you very much, Erica. Been great having you on. And
My pleasure.
Thank you all.